Okay, so as you know, or I'm hoping that you know, our current unit is on DNA and biotechnology. And what I'd like you to do is right now in your notebook, go ahead and at the very top of the page write Unit 1, DNA and Biotechnology, just like you see on this slide. And also go ahead and write down the area of interaction, which is health and social education, and our unit question. So this will be sort of your setup page as we begin to take notes on some of the background information that you'll need in order to complete future labs during the course of this unit and to understand some of the bigger questions that we're going to wrestle with about whether or not it's a good idea to clone animals, whether or not we think that genetic engineering of our food is a good idea, all those questions. In order to answer those, you need to have some basic background information. So at this time, go ahead and pause this recording and set up your notebook. And remember to put the date in the left-hand margin. So put today's date in the left-hand margin and then write this information from the slide in your notebook. Okay, so hopefully you're back to play. You've hit pause, you've set up your notebook, and now you've hit play. So I just want to tell you that making a screen recording is scary for me. I don't like it. I don't really like lectures in general, and I know some people learn really well from a lecture, and other people do not. So those of you who do not learn well from a lecture, I'm asking you to be patient because we will have some hands-on activities to supplement this information. And just go ahead and push through it, pause when you need to so that you can digest the information. And I'd also like you to, at this time, you're going to pause this recording one more time, and I'd want you to set up a page, maybe it can be the next page or a separate page somewhere that just says questions. And if there's anything that you're really confused about during this screencast, go ahead and write those questions down, and that way we can come back to them and make sure that we clarify for you what you don't quite get. Like if you're not really sure, you still don't really get what a nucleotide is, or you're just really confused on what counts as biotechnology, write those down throughout this recording so that we can come back to them and it doesn't just get lost, and then when it comes time for the test, you're really in a bad place. Okay, so if I mess up, I'm just going to keep going because I need to just accept that I'm not perfect, and I hope you guys will just forgive me if there's anything that that really doesn't make sense. Okay, so here we go. Anytime you see this picture, okay, I admit it, it's cheesy. It's a wolf paw with a human hand. I think it's really cool. I mean, seriously, look at how big the wolf paw is. Anyway, when you see that picture, it means to pause. And you're going to pause the recording and do whatever it says on that page. So right now, pause and follow the instructions. Okay, hopefully you're back now. You've hit play again. And I'm hoping that you gave some examples of both positive and negative consequences. So it's not just that when we create new technology that there's negative things that happen, right? There's a lot of positive things that go along with our new understanding, whether we're talking about cell phone technology or in this case when we're talking about biotechnology. And we will share our answers to that question later on. So what is biotechnology? According to the United Nations Convention on Biological Diversity, so it's written right here, it's basically any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for a specific use. It's kind of a lot of mumbo jumbo, right? So that's your official definition. But what that means is when we use our understanding of life and we actually use life in some way to help ourselves, to solve our problems. So here's a really good example. Up until the 1980s, insulin was actually made from pigs or from cows. And insulin, what that is, you've probably heard of it before, but insulin is this internal secretion from the pancreas, and it's it's a hormone that we need in order to make glucose that provides energy for us. And most people right now, all of us watching this, unless there's somebody you don't know about, we make insulin. Our body makes insulin, and it keeps our blood glucose at acceptable levels. But some people that have type 1 diabetes, and that's the type of diabetes that you're born with, right? You don't just, there's some diabetes that you get because of your diet. But if you're born with type 1 diabetes, your pancreas is not creating insulin. And the cells that produce that insulin have, for some reason, been destroyed. And that has to do with your DNA. There's a mutation in your DNA that's causing this problem. So up until the 1980s, what we did to solve that problem for people that couldn't make insulin is we took insulin from the pancreases of cows, cattle, and pigs. And it's almost exactly the same as human insulin. Pork, or porcine or pork insulin has one different amino acid in their insulin than we do, and bovine, or from cows, they have 3 different amino acids. So it's very similar, and we were able to use it to help people create, or to help people have insulin that didn't have it. Yeah. So that's biotechnology. We used the insulin of another animal, and we used our understanding of our own insulin to make insulin for people that didn't have it. But now, after the 1980s, our understanding of DNA became more sophisticated, and now we actually make insulin in laboratories biosynthetically by recombinant DNA. We're basically making it ourselves in labs using our understanding of how bodies make insulin to begin with. So we've discovered the recipe for human insulin, and that recipe is locked in our DNA. And that's what this unit is about, is understanding how the heck does our body make insulin? Just like how does it make your fingernails, or how does it make your hair, or how does it make your skin or your eyes? Another example of biotechnology is this thing called the golden apple. It caused quite a controversy in Washington. And maybe some of you or your parents know about the GMO debate in Hawaii right now surrounding Monsanto. And we're going to talk more about that in the unit, but to give you another example, this golden apple, you'll notice the apple on the left is kind of brown. And you know that when you cut an apple and you leave it out in the air, it turns brown. And that's because apples have an enzyme that starts to break down the sugar in the fruit when it's exposed to oxygen. So it makes it brown. It's not bad. It's, a brown apple is still delicious. But what researchers figured out how to turn that enzyme off, how to make it not break down the sugar, so that when you cut up an apple, it doesn't turn brown. And they were thinking, consumers would like that apple better because, a lot of people get freaked out when their apple's brown. So they genetically engineered the golden apple so when you cut it open, it doesn't turn brown for a very long time. And of course, people are very mistrusting of this. They're wondering, is this good? Is this right? Should we be messing with this? And that's caused, again, a lot of controversy. A lot of farmers are like, we don't want this golden apple, right? That's another example of biotechnology. We're using our understanding of apple DNA to change the way an apple looks. So we have 2 different examples right here, and there's tons of examples of biotechnology. But one example, again, is the insulin. Another example is the apple itself. You could even go back even farther in time, and you could think of, when people first started making beer and wine, that was biotechnology. They're using yeast, which is a living organism, a little microorganism. They're using it to help break down the grapes and in the fermentation process, and in the brewing process of beer, they're using yeast to help them make beer or to make wine. So that's biotechnology. We're using a living organism to solve our problems. Or cheese is biotechnology. We're using mold to make a delicious food, right? So you could get very technical with modern biotechnology where you're actually going into DNA, or you could, talk about more basic biotechnology where you're using yeast or mold to make a food. Okay, so this brings us to cake. And you might be wondering, Ms. Ho, what does this have to do with what you just told us? First of all, cake is delicious. Let's just take a minute to acknowledge how delicious cake is. Second of all, anybody who bakes, and if you look at all these different cakes in the picture, which by the way, this is from a little bakery in Brooklyn that I visited last year. But if you think about cake, right, you follow a recipe to make it. Yeah, so maybe you have a couple cups of flour, you have some sugar, you have some egg whites that you had to beat until they were foamy, and then you added them in a certain order, right? You baked it for a certain amount of time. There's a recipe for making each of these cakes, and each recipe is a little bit different. It has a different ingredient, or it might, maybe you need to bake this cake for a little bit shorter period of time. And that actually, if you can just always remember this throughout this unit, that is the same idea behind DNA and living things. It's a recipe. So instead of cake, we look at a tree. We might wonder to ourselves, how do you make bark? What in the world is bark? What are the ingredients for bark or for the needles on this cedar tree, which, by the way, this picture is from Glacier National Park in Montana. Woot woot. But the question is, how are these made? Why did they grow so tall? What is telling them to grow taller? How are their branches made? So what's the recipe? Just like cake, what's the recipe for trees? Or we could go here, and if you look at each of these pictures, it's a living thing. What's the recipe for a toad? What's the recipe for butterfly wings, or for a kitten, or for a human baby, right? And I think actually the human baby one might be one of the most important questions, because you have this creature growing inside of a mom's uterus for 9 months, and you're like, how is this thing growing? Like, what is it? It's an alien. Then it comes out, and over time, it grows into a toddler, and then into a young kid, and then into a teenager, a sassy teenager, and then into an adult, and then over time into an old person. So what's happening? How is that? What's going on? And I think that if you think about science, it always starts with curiosity. And humans are looking at all the life around them, and they're wondering, where did this come from? How is it made? And I'm going to give you an example. Up there in the left-hand corner is my friend Jocelyn. And in my humanities class, we talked about her mom passed away last year from a type of cancer called lymphoma. And this is where our understanding of DNA becomes really pertinent to our lives, because as we know, a lot of people these days are getting cancer. And the question is, what is that? Okay, so lymphoma. You have your lymphatic system, which is a part of your immune system, and it consists of all these vessels, that carry a fluid that's called lymph. And it's like a blood vessel carries blood, but instead your lymphatic system carries lymph. And lymph contains the white blood cells, and we have our B cells and our T cells, So the B cells produce antibodies that attack a foreign cell and alert your body of invaders. So let's say all of a sudden you get a virus, your B cells are like, attack, we've got invaders. And then your T cells are the actual army fighters. They charge them. They directly attack the pathogens. So what happens, it's a really cool system actually. You have your own army. Each of us has a lymphatic system, and anything that invades our body, whether it's a bacteria or a virus, our B cells and T cells in our lymph attack it. So they alert our body, and then it's a massive battle. And usually we win, right? You guys all get colds, and you win. It might take you down for a couple days, but you win in the end, because your body is a rock star at fighting them off. Well, in lymphoma, what actually happens is those lymph, the B cells and T cells actually start to grow and multiply out of control. So normally they multiply, the old cells die and new cells come into place. But when you have lymphoma or any type of cancer, what it is what we call radical cell growth. All of a sudden, if you have lymphoma, your lymph cells start growing out of control, and they form tumors. And those tumors, which are just a bunch of extra cells that don't belong there, they take up space, and they cut off the supply of oxygen and nutrients to the other cells. So all of a sudden you have like this big blockage in your system, and your body can't fight off viruses and bacteria. And so you die from a common cold. And that's actually really scary when you think about it. But the reason this relates to our unit is, the question is, why were those cells multiplying out of control? What happened? Why all of a sudden did these tumors form? And what that is there was some message in Jocelyn's mom, her mom's name is Hailuan, in her DNA that told those cells to multiply out of control. And where was, so something happened to her DNA that caused a message, the wrong message, to tell her body to create extra cells, and then from that she died. So as we research DNA, the more we know about it, the more we can figure out how to tell that cancer to stop. We can actually go in the DNA and be like, hey, turn that off. We don't want that radical cell growth. Okay, the same thing with any of these creatures on this page. It's basically inside of their bodies and in the middle of their cell is information on how to create them. And when things go wrong, then we can go back into that information and try to make it right again. So the key word is cells. Cells are the building blocks of all life, whether it's plant life or a little tiny snail or a human being. And you can think of cells like bricks. If you look at this picture here, it's the Alhambra, which is a very famous building in Spain. And it's incredibly intricate, and it's very old. It's over 500 years old. And but what's interesting about it is that it's made of bricks. I mean, essentially, it's a bunch of bricks put together in a pattern to hold up this huge fortress. And that's no different than your body. Your body is made up of little bricks called cells, and those cells lock together to form this intricate human being. And you may have seen pictures of cells in the past, right? So on the right-hand side, I'm guessing that your brain just said, oh, that's a plant cell. I'm hoping it did, because you should have learned it in 7th grade. And a plant cell, you can tell right away because it has the plastids, which carry chloroplast. So it has that big green thing going on there. Whereas on the left, you have an animal cell. And you know that both of these are animal and plant cells because they have a nucleus, right? That big blue thing, if you can look at it right now. And the nucleus is the command center for your cells. And the key piece here is that quote at the top, every cell comes from another cell. It's really important to think about. Every cell comes from another cell. We replicate our cells. Right now, as I'm talking to you, your cells are replicating, and then they're rebuilding parts of your body right now, as we speak, right? And so the question is, how are they replicating? What in the heck is going on? So sometimes, like let's say you fall and you get a cut, so your cells rebuild. They repair. That's how come that cut, eventually is no longer, there's no evidence of it. Or let's say you get a sunburn and your skin makes new skin cells, right? And so if we go back here, that's how a baby is formed actually. The chromosome pair, if you look up here, that's my friend Segalik, that's her little baby. Akatuk is her name. She's an Eskimo child. But basically what happened with that little baby is the chromosomes separated, they replicated, and they realigned over and over and over again. And that's how you get a human being, right? So every cell comes from another cell. Which brings us to our classic picture of DNA. And I know you've all seen it, and I know you've all heard it, the double helix, right? Let's take a look here on the right-hand side, and you see this double helix picture of DNA. And if you go backwards, if you can start at that kind of X shape, that is a chromosome. And all a chromosome is, I'm sure you've heard the term, but really a chromosome is just a bundle of DNA. Okay? It's just a bundle of DNA wrapped up really tightly around a protein called a histone. And if you unravel a chromosome, you actually have about 6 feet, 2 meters of this long molecule called DNA. That's what you have. If you unravel you can see here, if you trace to the right, it's basically showing you unravel that chromosome that looks like an X, and what you have is this long strand of DNA. And what you have in your DNA, this molecule, what's holding those 2 strands together is what's called the base pairs, A, T, C, and G. So you can see, if you take a look at the picture, the yellow is representing the A, the blue is representing the T, the red is the C, and the purple is the G. So you can see them, they're like locked together, okay? And A, T, C, and G are the first letters of what we know as the base pairs. So adenine, thymine, cytosine, and glycine, okay? So those are your 4 base pairs. And basically, all your chromosomes, if you go backwards in the picture now and you look at your cell, in your cell, in your nucleus, is where your chromosomes are stored. And that's where your DNA is. Yeah? So if you just kind of go backwards, you're like, okay, I have this strand of DNA that bundles up to make a chromosome that lives in the nucleus of my cell, and the cell forms tissue, or it forms your eyeball or your organs, right? And those cells are constantly being copied and reformed. Okay? So I'm gonna give you time to take notes on this, but I just want you to take a look at the picture and kind of picture this is what we have going on in our bodies. That's why these cells can replicate. Okay. Let me go forward. So here's like an analogy that helps me is if you think of a bunch of spools of thread, that's kind of what a chromosome is. A chromosome is basically, like I said, it's a bundle of DNA, and it's wound really tightly. So each one of these spools of thread kind of represents a chromosome. It's a bundle of DNA, a long strand wound really tightly around a histone. Okay? And humans, as you know, we have 23 pairs of chromosomes. So we have a total of 46 chromosomes, one from our mom and one from our dad. So if I go back to this slide, this X, this chromosome, let's pretend that that's chromosome 11, right? So this might be the copy from your mom, and then you have an identical one, or a very similar one, chromosome from your dad, right? So chromosome 1, there's 2 of them. Chromosome 2, 2 of them. Chromosome 3, 2 of them. All the way up to chromosome 23, which is your sex chromosome. So if you're a boy, you have 1 X and 1 Y. And if you're a girl, you have 2 X's for chromosome 23. Right? So you kind of have all these bundles of DNA wound really tightly together, and each one of those bundles has information on it about your genes. Right? So I think your eye color is on chromosome 18 and I think chromosome 5. So you have 2 different genes that kind of pair together to determine your eye color, and they're located on 2 different chromosomes. So if you're looking at these bundles of thread, or these spools of thread, you could picture maybe the pink one is chromosome 18, and maybe the sort of teal colored one over here is representing chromosome 5. And they have different genes on them. On these bundles of DNA, there's different information on each of them. So right now what you're going to do is, and if you need to back up and listen to something again, you should. But go ahead and pause at this time. You see my pause, and follow the directions. Okay, hopefully you're back and you've followed the directions and you've written down your notes, and you're welcome to add more notes if you need to. So the question is, what happened? How do we even get this information to begin with? So here's the history that you need to know. It's kind of anybody that's taken a biology course knows these names, and you should know them as well. So a group of scientists working at Cambridge University in England was working, they're kind of in this like race to find out really what is DNA. They couldn't see it, right? Because DNA is smaller than a light wave. And so if something's smaller than a light wave, you can't look at it under a microscope. You have to find a different way to look at it. And so this woman named Rosalind Franklin, she decided to start taking x-rays of DNA to get an image. And she got this image right here. And as you can see in the center there's this X-looking thing, which happens to be looking at like the chromosome. And what happened is, because she was taking all of these x-rays, she was exposed to a lot of radiation, and it caused her to develop ovarian cancer, because radiation actually breaks down your DNA and causes mutations. And so she died before her photo could really, she could get full credit for it. But her colleagues, Watson and Crick, they used that photo to then, we'll go to hers. Over here, here's Watson and Crick. There's a picture of them, and Rosalind Franklin's in the middle. They used that photo of hers to make a stick and ball model, which you can see here in the background, of DNA. And they used a lot of math to figure this out, right? And they actually got the Nobel Prize for figuring it out because it has completely changed our understanding of life. And obviously the medical field has been changed significantly because of our understanding of DNA, and as has agriculture, our production of food, our understanding of animals. So much has changed with this new understanding of the structure of DNA. So Watson and Crick, you should know their names. They're the guys that are credited with developing the full structure, but they couldn't have done it without Rosalind Franklin. Okay? And you know what? I just realized when I was telling you those base pairs, I think I said glycine, which is actually an amino acid. So sorry about that. It's guanine. We'll get to that in a second. Forgive me. Okay, so you're going to go ahead and write this in your notes, and you're going to pause and make sure that you just have this basic information about who discovered the structure of DNA. Just because, you know, it's one of those historical pieces of information that everyone should know. Okay, so hopefully you're back, and we're going to move on. So, DNA, the acronym DNA stands for deoxyribonucleic acid, right? And what that really is, the nucleic acid part of it, is related to nucleotides. And I'm going to give you a chance to write this in your notes. So, if you look at that double-stranded molecule that we looked at a picture of before, and I told you that there were those bases, right? Those 4 bases in the middle. So DNA is made up of nucleotides, these little kind of, how would I say it? Like these little, almost like Legos, if you could think of Legos. Each block is a nucleotide. Okay? And the nucleotides have 3 basic parts. They have a sugar, they have a phosphate, and then they have the base that we talked about before, the ATCG. Okay, so that's what a nucleotide is, and that is the building blocks of the DNA molecule itself. So when, I'm going to go back here, whoa, back. Okay, so if you look at Watson and Crick's structure there in the background, you can see that they built with these sticks and balls, and you're going to actually do that here in a little bit. What that's made up of is those nucleotides. Each of those stick and ball models is made up of a phosphate, sugar, and a base. Okay? And by the way, Watson went on to launch the Human Genome Project, which is mapping all of the genes that humans have. And that's how you can like take a swab of your cheek and send it in. You send in a little bit of your DNA, and they analyze your DNA and tell you like what diseases you might have a better chance of getting, or that you maybe have no chance of getting. And we'll learn more about the Human Genome Project later in the unit. Okay, so here we go. Deoxyribonucleic acid is what DNA is, and it's built up. It's made up of nucleotides, like these little blocks, building blocks of DNA. And each nucleotide is made up of a sugar, a phosphate, and a base. Okay, so I'm going to just move more quickly. Okay, so here's what you're going to draw this in just a second, but I'm going to show it to you first. Don't be thrown off by any of this fancy language either. Just think about water. Water is a molecule, you know it, H2O. It's 2 hydrogen and an oxygen, right? So it's atoms bonded together. So all we're doing now is we're looking at a different kind of molecule, not a water molecule, but a DNA molecule. We're looking at what it's made up of, okay? So don't get scared by some of this fancy chemistry language. The monomer and the polymer, it's all it is just telling you the ingredients of DNA, okay? So you have your phosphate group right here, which is made up of phosphorus and oxygen. And then you have your sugar right here in the middle. And then the last piece, you have your nitrogenous base. And the reason it's called a nitrogenous base is because it's made out of, part of its basic ingredient is nitrogen. Okay? So that right there is a nucleotide. That picture is a nucleotide. And you'll have a chance to totally draw this in just a second, okay? So here you have your sort of twisted ladder known as the double helix. And the sides of the ladder, if you can see the blue, like kind of ribbon looking thing, those sides are what's called the backbone of your DNA, and that's your sugar and your phosphate, okay? And then the middle part of your DNA is your ladder rungs, and those are your bases, okay? So just take a second and look at it. So the backbone part of your DNA is the phosphorus and the sugar, the phosphate group and the sugar. And in the middle, the rungs that kind of click into place and hold the whole thing together are your bases. Okay, so those are the nucleotides. So if you look there at the top of the DNA and you see the A and T, so the A is connected to a sugar and a phosphate group. Excuse me. So that's one nucleotide. And the T is connected to a sugar and a phosphate group, and that's another nucleotide. And then they lock into each other and hold DNA together. Okay, so this I think will be a little bit easier to see. So there are basically two rules for how DNA fits together. Adenine always pairs with thymine. So the A and the T are always paired together. And you can see their chemical structure, but I'm not going to make you memorize that, so don't write it down. You don't need to know it. You just need to know that thymine and adenine always pair together, and those are your bases. Those are your nitrogenous bases. And guanine and cytosine always pair together, and those are your two other nitrogenous bases. Yeah, so your bases are A, T, C, and G. You just kind of got to get used to those four letters because they're going to come up a lot this unit. Adenine, thymine, guanine, and cytosine. And so if you look to the right at the double helix, again, you'll see the A's and the T's are always paired together, and the C's and the G's are always paired together. And we call those complementary pairs. So this is your chance to actually write some of this stuff down. And if you want, you can go back back a couple slides to look at the actual pictures, whatever you need, but you're at this time going to go ahead and pause the screencast and you're going to take notes to make sure you have all this information. Okay, so hopefully you're back. So I'm going to give you this again, this slide that you already saw, and I'm going to ask you to go ahead and draw it in your notebook. So you're going to pause on this page, and I didn't put a pause on this page, I'm sorry. I messed up. There's no wolf and man, giving each other a high five. But go ahead and draw this. This is your nucleotide. So go ahead and, you don't need to write all the words. I want you to draw the phosphate, the sugar, and the nitrogenous base, and then label it as a nucleotide. And this is basically, again, remember the phosphate and the sugar make up the backbone. And then the base over here, the nitrogenous base, is your ACTG. That's what the green is. Okay, so go ahead and draw that for yourself because you will need to know this basic structure. If I ask you to draw a DNA structure, I would expect you to draw, if I asked you to draw a nucleotide, I would expect you to draw this. Okay? Okay, hopefully you're back. So, let me just go for it. This right here, on the right-hand side of this picture, is a really good way to look at how DNA is held together. So you see that pentagon, which is your sugar, and the circle, which is your phosphate, and then it's linked onto a base, right? So the next slide is going to show you an actual closer up picture of this, and you're going to draw it in your notes. But this is, again, that same idea. Each of these three components, the circle, the phosphate, the pentagon, which is the sugar, and then the base, that represents one nucleotide. And then you can see that each nucleotide is linked together to form this long chain, which then becomes basically DNA. Okay? So here you go. Go ahead and draw this in your notebook. And you don't need to draw all of them. Just go ahead and draw maybe GCTA, just those first four. And draw your circle and your pentagon and your bases so that you have a good picture, and then label your backbone. The sugar and phosphate is the backbone. And then go ahead and label the GCTA, guanine, cytosine, thymine, and adenine. Label them with their names and write down that those are your nitrogenous bases. Okay, so I'm hoping that you've stayed with me so far. I know it's a lot to take in, and we are going to cover it again. This is just sort of your first go at it. And then when I'm back, we'll go over it again and make sure everybody gets it. But this is a way to really think of it, and I think it will help you. If you think of a cell, and remember I told you, your cells are constantly replicating. It's so weird to think about. Like, you could look at your hand right now, and what you can't see is that inside of each cell, each skin cell, is a nucleus, and inside of that nucleus is DNA, and it's replicating right now while I'm talking to you. And your cells are multiplying within your body as we speak. Your heart cells, your stomach cells. It's actually kind of gross, but it's really cool. So let's think of the nucleus like this giant library, okay? And then in that library, it's kind of like a bunch of cookbooks, okay? So this library is reserved especially for chefs. And all it is you have a bunch of cookbooks, and they're in pairs. So cookbook number 1, there's 2 copies. Cookbook number 2, 2 copies. Cookbook number 3, 2 copies. Yeah, you get it. So as you could guess, each cookbook represents a chromosome, okay? So the nucleus is the library, and that's where your DNA lives. So if I ask you, where is your DNA located, you better know it's located in the nucleus of your cell. It's on lockdown because it's the most precious information that your body has. Okay? So you have this library with all these cookbooks. And how many cookbooks do you have? Should have said 46. You have a total of 46. Yeah? 23 pairs. So those are your chromosomes. Okay. So humans are diploids. Yeah? So that means that we have pairs. So the di prefix there is pairs. Just like strawberries are octoploids. They have 8 sets of each chromosome. So there, as you might guess, they have maybe more copies of each book than we have. Okay? So here you have this library, and it has 23 pairs of books or chromosomes. Okay? And in those books, you basically have recipes. Okay? And this is where we're going back to cake. If I Google chocolate cake, there's hundreds of different recipes for chocolate cake, and they're changing a few recipes, or they're changing a few, like the order that you put in the cinnamon and the sugar, or the chocolate, or maybe you drizzle it on at the very end, okay? And so each recipe is a little bit different. So we can think of that like your genes, okay? Whoops, let me go back. So each gene in that cookbook is a recipe, okay? So again, we have a library in the nucleus, okay? And in that library there are books, which are chromosomes, and in each book, in each chromosome, there are genes, and those genes are your recipes. When you look at all the recipes of a human, it's called your genome. Yep, so that word in red, the genome, it's all of your chromosomes with every single recipe in it, is the whole thing together. If you were going to buy them all together, it's called your genome. And the Human Genome Project, which was completed in 2002, the goal was to sequence all of the DNA on all 46 chromosomes and figure out what each gene was for. And in reality, we just found out recently that we know very little. We thought we knew a lot. We actually know very little. But we do know certain things. We do know that like certain genes are linked to certain cancers. Or we know that certain genes are linked to, or like, for example, trisomy 21 is when you're going to have Down syndrome. We know that we can definitely tell that certain things are genetic. but we don't know the whole picture. We thought we did, but we actually know a tiny little pixel of the picture. So we're going to go ahead and I'm going to have you write down your analogy. And here it is written out for you. And when you're done with that, and if you want to go back to draw any of it, you can. And then you're going to push play. We only have a few more slides. But make sure you kind of understand, most importantly, that the DNA is in the nucleus, and that it's in little bundles called chromosomes. And we'll pretend that each of those chromosomes is a cookbook. And in each cookbook, there are recipes, and those recipes are called genes. Okay, so go ahead and pause. Okay, hopefully you're back. I'm going to give you one more example before I let you go, just to make sure that you get it if you didn't get it the first time. See those little purple circles? Those are your histones that we were talking about earlier. And that's how DNA bundles itself up really tightly, and then forming eventually the chromosome. So I just wanted you to see that's what they look like, and it's just like this super tight coil. Okay? And I want to show you this analogy one more time. If you didn't get it the first time, this is the time that you might even want to go ahead and draw this example in your notebook. So here you have the green thing, which is your cell. And inside of your cell is your nucleus. And inside your command center, or your big library. And inside of your nucleus are your chromosomes. So you can see in that picture. And when you unwind your chromosome, what you have is a long strand of DNA. And you can see it right there. There are your base pairs, your adenine, thymine, guanine, and cytosine. Yeah, with the backbone, if you can look at the picture, the backbone there, the sugar and phosphate groups holding it together. Okay, so there's your DNA and your chromosome. And if you do a close-up on your DNA, then you can see that one recipe, one little section of DNA, because genes are only on sections of DNA, but that section of DNA is a recipe. It tells your body how to make a particular protein. Okay? So that's what happens when your cells replicate. It's reading the genes and telling your body how to make certain proteins, like the proteins that allow your eyeballs to see. Okay? Oops. So go ahead and actually draw out the analogy if you didn't already. And so I'm going to go back one slide. This slide is really helpful. If you want to go ahead and draw it, it will make it easier for you to understand. So go ahead and pause. And hopefully you're back. Okay. So the last piece of information that you need. If you look down here, the code that we're trying to crack, and that we're going to be working a lot with throughout this unit, is written in 3 letters each. So we have those A, C, T, and G. Those are the only letters that we can work with. But when 3 of them are together, that counts as a word, and it's called a codon. Okay, so you can see right here, AAT is one word. It's a codon. And then you can see it repeated. Or maybe you could have CCG. That's one word. It's a codon. So make sure that you write down codon, and then in the next slide or the next 2 slides, I'll give you another chance to pause and write that in your notes. But we have this long strand of DNA, kind of like a never-ending story, and there's little sections of it that are recipes, and those recipes have combinations of letters that are called codons. And I think this is a good way to look at it. And what you might notice right away is a U, and that's because when DNA gets read in the nucleus, RNA, which takes the information out of the nucleus, it changes the T to a U. Okay, so don't freak out and get super confused. Just know that T and U are really the same. They change a little bit when it leaves the nucleus, okay? But I want you to go ahead and draw this, so you'll see that this is one strand, and if it were to lock together, it would have the complementary base pairs. But it shows you the codons, okay? So AUG is one codon, one genetic word. That's what a codon is. It's a genetic word. ACG, another codon. Yeah, and again, the A stands for adenine, The U actually stands for uracil, but its other name is thymine, and then the G stands for guanine. Yeah, so it's just what we talked about already. It's just now one more piece to the puzzle, which is that when you put these letters together, they form codons. And the codons are basically, if you can look at this table, and you look on the right-hand side, it has all these different codons, yeah? ATT, ATC, ATA, all these codons. And the codons code for amino acids, which you see on the left-hand side. So amino acids, if you go to the supermarket, there's all these different advertisements that say like complete amino acids, or like get your full amino acids with this like new superfruit. So amino acids are basically the building blocks of your proteins. So your DNA has the recipe, which is over here on the right. Yeah, these are the ingredients that you might see in your DNA, which tell you to grab, let's say, valine. Yeah, that's the third one down. And then maybe valine links on to glycine or proline. Yeah, so you form a chain of amino acids which make a protein. And again, we'll go over this in greater detail. This is your basic introduction to the information, so don't panic. So what I'd like you to write in your notes is that a gene is basically a recipe for a protein, and it's made up of codons. And again, codons are genetic words, and hopefully you drew this picture. I'm going back to it. You drew this picture right here in your notes, and if you didn't, make sure you do. And then go ahead and pause and write down this information. And then you're back, hopefully, and now you have what is a codon. And again, here's the example if you didn't get it the first time. If you did, you can just, quickly write down this information about a codon. And hopefully you're back. And here is your full analogy. Okay, so this finishes up all the information that you need for today. So a genome is all of your cookbooks. It's all of your chromosomes together. Okay, a chromosome is a single cookbook. Yeah, it's a bundle of DNA. Your gene is just one recipe in that cookbook. A codon is one word in the recipe, yeah? So if we're thinking of DNA, so a codon is just 3 of those bases in a row. So your bases, adenine, guanine, thymine, and cytosine, are the letters in your DNA. Your amino acids are what the codons tell you to get. Yeah, so that long chart of a different amino acids, that's what a codon tells you. So you could think of it like a recipe. If you read the letters Flour, you would know to take a scoop of white flour, right? So in DNA, if you read AUG, you would know that the amino acid methionine needed to be grabbed. So it's kind of the same idea. And then the protein, once you read a full gene, then what you have from that is a protein, which is like your meal that you're making. And if you have a mutation in your gene, it's like as if instead of reading FLOUR, somebody accidentally deleted the L and it just said FOUR. And you're like, what is that? I've never cooked with four before. It's supposed to say flour. So in DNA, if you were supposed to have methionine, it would be AUG. But if it accidentally said ATG or just AG, your body wouldn't know what to grab, and then you'd have a problem. So here's your concept check. So what are the 3 parts of a nucleotide, and which parts make up the backbone of a DNA strand? So see if you can answer that for yourself right now. The answer is the 3 parts of a nucleotide are sugar, phosphate, and a nitrogenous base. And the sugar and the phosphate make up the backbone. So list the 2 base pairs found in DNA. See if you can answer it. So you should have said adenine and thymine, so the A and the T, and guanine and cytosine, the G and the C. All right, and then the third question, if 6 bases on one strand of DNA double helix are AGT, CGG, what are the 6 bases on the complementary section of the other strand of DNA? So you might be like, what? But you know the answer because it's just the base pairs. So what pairs with the A? T. What pairs with the G? C. Right? So you can write it out. You can actually answer the code. What's on the other side is the DNA. And the answer is TCAGCC. So you can write those right next to each other and you'll see that they pair up. Right? So that's how they lock together. And that's it for today. And when you're done, I want you to just go back through, make sure your notes are well done. If there's anything you're super confused about, go ahead and write the questions in your question section. And then you need to meet with Ms. Mullen to know what your next step is.